Imagine running a blood test on your commercial layer flock.
Right.
And you see absolutely perfect immunity scores. Like the numbers look fantastic. So you assume your birds are completely protected.
You'd think so, yeah.
Exactly. But then you walk into the laying barn and you find that egg production has just inexplicably plummeted. I mean, the eggs that are actually there, they have these incredibly thin, deformed, or, well, completely missing shells.
The nightmare scenarios.
It really is. And it brings up a huge question. Why would farm managers see perfect blood test results, but just absolutely terrible eggs?
Right.
Today, we are solving that exact paradox.
We really are. And it is a deeply complex biological puzzle.
Yeah, and to figure it out, we are digging into some really intense academic reports. We're covering the reproductive pathology of Newcastle disease, examining the molecular epidemiology of the virus circulating in Vietnam, and breaking down some, well, highly advanced dual-platform vaccine strategies.
That's a lot of ground to cover.
It is. But we are going to explore all of this at a very deliberate conversational pace. This deep dive is designed specifically for those of you out there looking to practice your English shadowing.
Right, while also mastering some professional poultry vocabulary.
Exactly. But make no mistake, we are treating you like the industry professionals you are. So let's just jump right in.
Sounds good. To really understand how to protect a flock,
Yeah.
I mean, we have to move beyond just reading basic vaccine labels.
Yeah, that doesn't cut it anymore.
No, not at all. We have to look at the virus on a molecular level, We need to examine the specific genetic mutations that make these modern strains so devastating.
Right. Like, what are they actually doing inside the bird?
Exactly. We will look at exactly how they shut down a hen's reproductive organs. And then we are going to evaluate the actual architectural blueprint of a modern vaccination program.
Specifically, we'll be contrasting those recombinant vector vaccines with the traditional oil emulsion-killed vaccines, right?
That's the core of it, yeah.
Okay, well, let us start by identifying the specific enemy we're dealing with here.
It's a tough one.
It is. Our sources focus heavily on the current threat environment in Vietnam. Newcastle Disease virus, or NDV, is just causing massive economic disruption over there.
Yeah, the reports isolate the specific strains causing this damage. And they belong to genotype 7th.
Genotype 7th. And if we dig even deeper, we are looking at subgenotypes 7.1.1 and OTSIV.2.
Right, and these are classified as velogenic strains.
Velogenic.
Yeah.
Which basically means they are highly virulent, right? Like they cause severe rapid disease.
Exactly. They move incredibly fast.
Let us actually pause on that classification for a moment.
Yeah.
Because it introduces our first key vocabulary word for our shadowing practice. The term is genotype.
Genotype.
Right. A genotype is the genetic makeup or, well, the specific family group of the virus. To say that again slowly, genotype.
It's a fundamental concept to grasp.
It is. And if you were walking the barns, a practical poultry farming example you might use or hear is this. The farm had a vaccine for one genotype, but a completely different genotype of the virus attacked the flock.
That happens all too often, unfortunately.
Yeah. So looking closely at the biology of these genotype the 7 strains, their devastating power comes down to a very specific genetic mutation.
It does. It occurs at what we call the fusion protein cleavage site.
The fusion protein cleavage site, okay.
Yeah, and in the genetic sequencing, this specific mutation is marked as 112RRRKRF117.
Wow, okay. That is quite the sequence. 112RRKRF117.
I know, it sounds like a password. But while you do not need to memorize the exact amino acid sequence, it's crucial to understand what the sequence actually does mechanically.
Right, like how does it operate inside the bird?
Well, this specific arrangement of basic amino acids acts like a biological master key.
A master key.
Yeah. Think about a mild virus. A mild virus can only be cut open and activated by enzymes found specifically in the bird's respiratory or intestinal tracts.
So it's very localized. It can't really spread too far.
Exactly. But with this velogenic 112RRRKRF117 mutation, the virus can be activated by ubiquitous enzymes.
Ubiquitous meaning they are everywhere.
Right. Enzymes found in virtually every single tissue in the bird's body.
Oh, wow. So that changes everything.
It really does. This master key allows the virus to rapidly fuse with host cells, multiply explosively, and just destroy tissue systemically across multiple organ systems.
So it's not just a respiratory bug anymore. It's everywhere.
Everywhere. And that systemic explosion is exactly what sets up our primary paradox.
Ah, right. The good blood, bad eggs problem.
Exactly. Farm managers are, actively trying to protect their flocks against these villagenic genotype 7 strains.
Sure, they're vaccinating like crazy.
Right. They vaccinate the birds, and then they draw blood to test the antibody levels, which is standard practice.
Right. And standard testing often uses an older strain of the virus as the antigen in the lab, doesn't it?
Yes, typically a Lesotho strain. And the Lesotho strain actually belongs to genotype 7.
Okay, so they're using a genotype Zika antigen to test for protection against a genotype 6 virus.
You've got it. And when the lab results come back, they usually show extremely high hemagglutination inhibition titers. We commonly refer to those as HI titers.
HI titers, right. And the managers are seeing results like 8 to 9 log 2, which is huge.
Massive. On a practical level, an 8 or 9 log 2 means there is just a super high concentration of antibodies circulating in the bird system.
So the manager looks at that paper and reasonably believes, well, the flock is totally safe.
They do.
Yeah.
Yet, almost overnight, egg production drops by 15 to 20%.
Just off a cliff.
Yeah. And the shell quality completely deteriorates.
So to unravel this, we really have to track how the virus physically moves through the bird's body.
We do. And it actually uses the bloodstream as a highway. It uses it to reach the internal organs.
The bloodstream as a highway. That systemic spread actually introduces our second vocabulary word for today. The term is viremia.
Viremia, yes.
Viremia simply refers to the presence of viruses in the blood, allowing the infection to travel throughout the body. To repeat that term slowly, viremia.
It's the moment the virus goes systemic.
Exactly. And for a practical poultry farming example, you would say, once viremia occurs, the virus travels through the blood to the hen's reproductive organs.
That's exactly what happens. And the primary reason this viremia happens, despite those high antibody test scores, is something we call genotype mismatch.
Genotype mismatch. Let's break that down.
Sure. The farmer vaccinated the birds, and the birds' immune systems successfully produced millions of antibodies.
Right. The 8 or 9 log 2, the guards are there.
The guards are there. However, the vaccine was manufactured using a genotype Tuvis strain. But the virus attacking the barn is a genotype Civis strain.
So they belong to entirely different genetic families.
Exactly. So when the genotype 7 virus enters the bird, the genotype 7 antibodies rush in. They recognize it as a threat and attempt to grab it.
What?
But because the virus looks slightly different, they bind to it very loosely.
Ah. So their physical grip on the virus is weak.
Very weak. And because this binding is so loose, a small but critical amount of the genotype 7 virus manages to slip away.
It just wiggles right out of their grip.
Yes. It escapes neutralization entirely and enters the bloodstream.
And boom, there's your viremia.
Boom. This creates a viremia breakthrough, allowing the virus to ride the circulatory system directly into the highly sensitive reproductive tract.
Okay, I have to push back on this entire strategy. I mean, if I am managing a commercial operation, I'm looking at this and thinking it's crazy.
How so?
Well, if we know the vaccine is a genetic mismatch, and we know the virus slips right through the weak grip of the antibodies, why use that vaccine at all?
It's a fair point.
Right. It seems incredibly counterproductive. Why wouldn't the industry just immediately pivot to using a genotype 7 vaccine that matches the field virus perfectly?
That is the logical next question. And the answer relies on a very strong foundational scientific consensus in veterinary immunology.
Okay, lay it on me.
Well, despite having different genotypes, all strains of Newcastle disease virus actually belong to one single serotype.
One single serotype, okay.
Yes. This serotype is called avian paramexovirus type 1, or APMV1.
APMV1. So they are fundamentally related at a base level.
Exactly. And because they share this underlying serotype, cross-protection is biologically possible.
Oh, I see.
A high-quality genotype 7 vaccine can still completely neutralize the genotype 7 virus, but only under one strict condition.
What's the condition?
It must create a supersaturated concentration of antibodies.
So you just need an overwhelming amount of them.
Precisely. If the blood is absolutely flooded with antibodies, the sheer overwhelming volume compensates for the weak, loose grip.
Like having 10 weak guards hold down one intruder instead of just one strong guard.
That's a great way to put it. The immune system basically swamps the virus by purer numbers, neutralizing it before viremia can occur.
But there's a risk there, right?
A huge risk. If that antibody titer drops even slightly from that supersaturated peak, the weak grip is exposed, the virus escapes, and the bird is compromised.
So it is essentially a high-stakes numbers game.
Completely.
You are relying on an overwhelming sheer volume of guards to compensate for the fact that they aren't, the best trained guards for this specific intruder.
That's the reality of the current vaccine landscape.
But let's look at what happens when those guards inevitably fail. When the viremia breakthrough happens and the virus reaches the reproductive tract, what exactly is it doing to the egg factory?
The pathology is incredibly severe, and it targets specific sections of the reproductive anatomy in totally different ways.
Okay, let's trace it. Where do we start?
We have to start at the top of the reproductive tract, which is the ovary.
The ovary, where the yolk is made.
Right. This is where yolk development occurs. When the Newcastle disease virus reaches the ovary, it immediately attacks the granulosa cells.
And what does it do to them?
It infects them and triggers a biological self-destruct mechanism within the cells themselves.
A self-destruct mechanism. Wow. And that actually brings us to our third vocabulary word for our shadowing listeners. The term is apoptosis.
Apoptosis, yeah.
Apoptosis is programmed cell death, or, when a cell is triggered by a signal to literally destroy itself from the inside out. Once again, that term is apoptosis.
It's basically cellular suicide.
Yeah, it's brutal. A practical poultry farming example for this would be the virus caused apoptosis in the ovary, meaning the cells destroy themselves and the hen stopped laying eggs entirely.
And beyond just forcing those local cells into apoptosis, the virus induces massive collateral damage through the immune system itself.
The immune system makes it worse.
Much worse. The infection triggers what we call a cytokine storm.
A cytokine storm, I've heard that term.
Yeah, it's when the dying cells release a massive surge of inflammatory signaling proteins. In this case, specifically interleukin-6 and tumor necrosis factor alpha.
So IL-6 and TNF-alpha.
Right. And this is not just local inflammation in the ovary. These proteins enter the blood and travel all the way up to the bird's brain.
Wait, all the way to the brain?
Yes. Once there, they interfere with the hypothalamic pituitary axis.
So it's messing with the brain's control center.
Exactly. They essentially block the brain from sending out the vital reproductive hormone signals.
Which hormone?
Specifically, luteinizing hormone and follicle-stimulating hormone, or LH and FSH.
Okay, so the brain just stops telling the body to make eggs.
Without those hormonal signals from the brain, the ovary undergoes severe regression. The entire yolk-forming process just shuts down completely.
That is devastating.
It gets worse. The hen's serum phosphorus levels plummet concurrently as a result of all this.
So the very top of the production line, the yolk factory, is completely shut down.
Totally offline.
But a hen usually has several eggs already moving down the biological conveyor belt, right? Like, there are eggs already in process. What happens to the eggs that are already past the ovary?
Good question. Those eggs move into the magnum, which is the thickest part of the oviduct.
A magnum.
Yes, and the magnum is entirely responsible for synthesizing the albumin.
The albumin, which we know as the egg white.
Exactly. When the virus infects the tubular glands of the magnum, it actively suppresses the production of ovalbumin mRNA.
It just turns off the blueprint.
Right. This mRNA is the essential genetic blueprint required to manufacture the egg white proteins. But the damage does not stop there.
It's supposed not.
The bird's immune system detects the infection in the magnum and sends killer T cells rushing into the tissue to clear the virus.
So the guards show up, but they end up breaking things.
Exactly. This intense cellular immune response causes severe local inflammation. It physically destroys the delicate glandular tissue.
So the glands that make the egg white are just wrecked.
Wrecked. And as a direct result, the glands can no longer synthesize thick albumin. You end up with what farmers call watery albumin.
Watery albumin. So if you crack the egg onto a flat surface, the egg white just sort of runs everywhere.
Yes, it's completely flat and runny, which correlates to drastically lowered HI units.
HI units being the primary metric used to measure internal egg quality, right?
Right.
Okay, so the yolk production halts, and the egg white turns into a watery mess. That leaves the final and, frankly, most visible stage, the shell.
Right, the shell formation.
Yeah.
This occurs in the uterus, which in avian anatomy is referred to as the shell gland.
The shell gland, what does the virus do there?
The virus executes a highly specific molecular attack here. It severely downregulates the mRNA responsible for expressing a protein called calbindin D28K.
Calbindin D28K. Okay, what is that?
It's a critical calcium binding and transporting protein. It functions essentially as a biological calcium pump.
A calcium pump?
Yeah, it pulls calcium out of the bloodstream and moves it into the shell gland fluid so it can crystallize into the hard egg shell.
Oh, wow. So when the virus shuts down the production of this pump,
the entire shell calcification process fails.
or it just stops.
Yes, this is exactly why you see shells that are paper-thin, severely deformed, wrinkled, or even laid without any hard shell at all, just a soft membrane.
That's horrifying to see on a farm.
It is. Additionally, the virus destroys the local pigment-secreting cells.
Oh, so the color changes too.
Right, meaning normally rich brown eggs emerge completely pale or white.
It is basically like a factory supply line. I mean, the virus isn't getting in there and physically smashing the eggs with a hammer.
No, it's much more subtle.
Right. It's sneaking into the factory and quietly turning off the specific machine that pumps the calcium concrete onto the egg. And because the pump has no power, the shell never gets built.
That's a perfect analogy. And building on that factory analogy, understanding this exact molecular mechanism changes how a manager should respond to an outbreak.
How so?
Well, when shell quality suddenly drops, the instinctive panic response for many farm managers is to heavily supplement the flock's drinking water with liquid calcium.
Oh, sure. They see thin shells, they think, give them more calcium.
Right. They assume a thin shell means the bird simply needs more raw calcium. But the pathology data proves this intervention is a complete waste of money during a Newcastle outbreak.
Oh, waste of money.
Completely. The bird likely has plenty of calcium circulating in its blood already. The issue is that the biological pumps in the shell gland are broken. The factory machinery is turned off.
Oh, I see.
So flooding the blood with extra raw material will not fix a broken pump. It just sits there.
That is a crucial insight for anyone managing a budget during an outbreak. Do not buy extra calcium.
Exactly.
So we understand how the virus uses the blood to break the factory machines, but, our outline mentions secret tunnels, which implies there's a completely different route the virus can take.
There is, unfortunately.
How else is this virus getting into the reproductive organs, if not through the blood?
Well, we have to remember that Newcastle disease is primarily a respiratory pathogen. It enters the bird via the respiratory tract.
Through breathing.
Right. Now, the internal anatomy of a bird is incredibly unique and differs vastly from mammals. We refer to this specific anatomical vulnerability as the lung oviduct axis.
The lung oviduct axis?
Yes. Birds possess a complex system of internal air sacs for respiration, and the abdominal air sacs sit in direct, intimate physical contact with the ovaries and the oviduct.
They are literally touching each other.
Essentially, yes. They are separated by incredibly thin membranes.
Wow. Okay, so how does the virus cross over?
When the virus infects the respiratory tract, the bird's immune system dispatches macrophages.
Macrophages, those are the big white blood cells, right?
Exactly. They are large white blood cells designed to literally swallow and digest foreign pathogens. So they swallow the Newcastle virus in the air sacs.
Like Pac-Man.
Like Pac-Man, exactly. However, these velogenic strains are incredibly resilient. They often survive inside the macrophage without being digested.
Oh no, so they just hide inside the cell that was supposed to kill them.
Exactly. The macrophage, unaware it has become a Trojan horse, then physically migrates across that thin membrane.
It just walks across.
It moves right from the respiratory system directly into the reproductive tract. It delivers the live virus straight into the egg factory, completely bypassing the bloodstream.
That is terrifying. It just uses the body's own security guards as a taxi service right through the secret tunnel.
It's a brilliant, if destructive, evolutionary tactic.
And this local, surface-level battle introduces our fourth vocabulary word. The term is mucosal immunity.
Mucosal immunity.
Right. Mucosal immunity is the localized immune protection found on the wet, internal surface linings of the body, such as the respiratory tract, the gut, or, as we just learned, the oviduct. Once again, that term is mucosal immunity.
It's the first line of defense on those surfaces.
Exactly. And a practical poultry farming example here would be the hen survived the virus thanks to blood antibodies, but she lacked mucosal immunity, so the virus still heavily damaged her respiratory tracts.
That happens quite frequently because this virus can exploit this secret anatomical tunnel, bypassing the blood entirely. Veterinary science dictates that birds require 3 highly distinct levels of protection.
Okay, let's break down these 3 levels. What's the first one?
The first level is simply mortality protection. This is about keeping the bird alive.
Just survival.
Yes. It requires a relatively low baseline of circulating antibodies in the blood, roughly an HI titer of 3 to 5 log 2.
So 3 to 5 log 2 keeps them breathing.
Right. With this level, the bird will survive the infection. However, the bird will still become severely clinically ill. And crucially, the virus will easily replicate in the respiratory tract and spread to other birds.
Spreading to other birds. This concept of the virus spreading from a vaccinated but sick bird leads perfectly to our 5th vocabulary word, The term is shedding.
Kidding, yeah.
Shedding is when an infected bird actively releases the virus from its body, usually through respiration or feces, into the surrounding environment, putting the rest of the flock at risk. To repeat that clearly, shedding.
It's the main driver of an outbreak in a barn.
Yeah, and a practical poultry farming example is the ultimate goal of the vaccine program is not just to keep the individual bird alive, but to completely stop the shedding of the virus into the barn environment.
Stopping that shedding requires the second level of defense, which is respiratory protection.
Respiratory protection. How do we get that?
To achieve this, you need a higher concentration of blood antibodies, an HI titer, greater than 5 or 6 log 2.
Okay, higher numbers in the blood.
Yes, but more importantly, you must establish strong local mucosal immunity in the respiratory tract.
The mucosal immunity we just talked about.
Exactly. You need a dense shield of localized antibodies, known as secretory IgA, lining the throat and the air sacs.
So they're just waiting on the surface.
Right. This local shield attacks the virus the moment it is breathed in, preventing it from replicating and, most importantly, preventing shedding.
Okay, so that's level 2. What's level 3?
Finally, we reach level 3, which is reproductive protection. This is the absolute gold standard for layer and breeder flocks.
The gold standard. How hard is it to achieve?
Very hard. Achieving this requires massive hyperimmunization in the blood, pushing HI titers up to 8 to 10 log 2 to completely seal off viremia.
So amass a wall of guards in the blood.
Yes. And simultaneously, it demands a dense, active mucosal immune shield maintained specifically within the oviduct itself to guard against any virus that attempts the air sac crossing.
Okay, let's visualize this lung oviduct axis as a secret underground tunnel connecting 2 heavily guarded fortresses.
I like that visual.
Right. You can hire 1 million heavily armed guards and put them in the bloodstream. If you pull a blood test, it looks perfect. Your fortress looks impenetrable.
The HI titer looks great.
Exactly. But if the virus simply walks through the unguarded air sac tunnel, it gets right into the egg factory. It bypasses all your expensive blood guards entirely.
That's the tragic reality for a lot of farms.
And this completely solves our initial paradox, doesn't it?
It really does.
A standard blood test, the HI titer, only measures the guards in the blood. It is completely blind to mucosal immunity. It cannot detect the presence or absence of secretory IgA on the surface tissues.
It literally cannot measure it.
So you can have a perfect blood test, but if your local mucosal shield is weak, the virus takes the tunnel and the egg factory is destroyed. The serology looks perfect, but the egg production is ruined.
That synthesis is precisely correct. The blood test tells you a true story, but it only tells you half the story.
Wow. Okay, so understanding this complex vulnerability, how do we actually go about building these 3 distinct levels of protection in the real world? It clearly requires a highly strategic plan.
It takes very careful planning, yes.
Let us analyze the first layer of defense, which involves what you call the vector vaccine breakthrough.
Yes. For modern commercial flocks, building this complex defense architecture begins on the very first day of the chick's life.
Day 1.
Right at the hatchery. Farm managers utilize a recombinant vector vaccine. The specific platform widely used is known as rHVTND, and it is administered subcutaneously to the 1-day-old chick.
Subcutaneously, so under the skin. Before we break down the exact mechanics of this vaccine, we have 2 related vocabulary words to cover.
Important ones, too.
Very. The 6th term is maternal antibodies.
Maternal antibodies.
Maternal antibodies are the temporary borrowed immune protection that is passed directly from the mother hen down to her chick through the egg yolk. To say it again, maternal antibodies.
It's nature's way of protecting the baby.
Yeah, and a practical poultry farming example is, the chick had a high level of maternal antibodies, which successfully protected it from field viruses for the first few weeks of its life.
That protection is vital early on.
It is. And that brings us right to our 7th vocabulary word, which is vector vaccine. A vector vaccine is a highly specialized vaccine that uses a safe, modified, harmless virus to carry specific genetic pieces of a dangerous virus into the body, safely training the immune system. Once again, vector vaccine.
It's an incredible piece of bioengineering.
Truly. And for a practical poultry farming example, we injected the day-old chicks with a vector vaccine at the hatchery to ensure they developed lifelong cellular protection.
Exactly. And to understand why this technology is so revolutionary, we need to look at how it is constructed in the lab.
How do they build it?
Bioengineers start with a completely safe, non-pathogenic virus native to turkeys. known as herpes virus of turkeys, or HVT.
So a harmless turkey virus.
Right. They genetically modify this safe turkey virus by inserting a single specific gene extracted from the deadly Newcastle disease virus.
Gene.
Specifically, the gene responsible for producing the F protein, or fusion protein.
The master key we talked about earlier.
A part of it, yes. When this engineered vaccine is injected into the day-old chick, it initiates what is known as a cell-associated infection.
Cell-associated? What does that mean?
This is a profoundly important mechanism. It means the vaccine virus does not float freely in the bloodstream.
Okay, where does it go?
Instead, it travels directly from one cell to the neighboring cell. It effectively hides inside the structural cells of the bird's body.
It hides inside the cells. If we unpack that mechanism, why is it so advantageous for the vaccine to hide inside the host cells? I mean, why not just be in the blood?
Because it operates intracellularly, it triggers a very specific branch of the bird's immune system. It activates the MHCI pathway.
MHCI pathway.
Which stands for Major Histocompatibility Complex, Class I. Activating this pathway generates a robust Th1 cell-mediated immune response.
Okay, let's strip out the deep jargon. What does that actually mean for the bird?
Stripped of the jargon, this means the vaccine prompts the bird's body to manufacture a massive permanent army of CD8 plus T cells, which we commonly call killer T cells.
Killer T cells. I like the sound of that.
They are very effective. These killer cells constantly patrol the bird's tissues for the remainder of its lifespan.
Just constantly on patrol.
Yes. If they encounter any host cell that has been infected by a virulent field strain of Newcastle disease, they recognize the specific viral proteins on the surface and immediately destroy the infected cell, halting the replication cycle in its tracks.
This sounds incredibly effective, but I have a major question regarding the practical application at the hatchery.
What is it?
Well, we just discussed maternal antibodies, the powerful guards the mother hen passes down through the yolk to protect the baby chick.
Yes, the MDAs.
Right. If those maternal antibodies are aggressively hunting for Newcastle disease in the blood, won't they immediately identify this vector vaccine and just destroy it the moment we inject it into the day-old chick?
That is a very logical concern.
Aren't we essentially wasting an expensive vaccine?
That exact concern plagued veterinary science for decades with older, traditional live vaccines.
Oh, really?
Yes, but this is the exact biological brilliance of the vector vaccine platform. We have to look at the physical location of these elements.
Location is key.
Absolutely. The maternal antibodies passed down from the hen circulate freely within the chick's blood plasma. They operate strictly outside of the cells.
Okay, they are out in the open.
Right. The HVT vector vaccine, however, utilizes intercellular bridges to spread directly from the inside of one cell to the inside of the next.
Oh.
Because the vaccine remains entirely sequestered inside the cellular walls, the maternal circulating antibodies literally cannot reach it.
I can't even see it.
Exactly. The vaccine safely evades the mother's inherited immune guards, successfully establishing itself and training those killer T cells without any interference.
That is a remarkable evolutionary workaround. It basically hides from the mother's protection in order to build its own independent defense system.
It's beautifully engineered.
But, as impressive as killer T cells are, they only represent one layer of defense, right? They seek and destroy infected cells.
Yes, they clean up after the infection is started.
Exactly. And for a long-living layer hen, we still need that massive, supersaturated wall of antibodies circulating in the blood to intercept the virus before it even enters the cells. How do we build those circulating guards?
You are absolutely right. For a laying hen, a Th1 cellular response is highly beneficial, but it is totally insufficient on its own.
They need more.
Much more.
Yeah.
Laying hens absolutely require a massive, sustained Th2 immune response.
Th2.
Yes. They need immense concentrations of circulating antibodies, specifically immunoglobulin Y, or IgY, which is the avian equivalent of IgG.
Okay, so massive amounts of IgY.
Right. They need this massive antibody wall to prevent systemic viremia over an exceptionally long production cycle, which can easily last 80 to 90 weeks.
80 to 90 weeks. Maintaining that level of antibody saturation for nearly 2 years is a monumental biological task.
It's exhausting for the bird system.
So what specific tool do farm managers use to achieve that long-term saturation?
They turn to inactivated vaccines, which are commonly referred to as killed vaccines.
Killed vaccines.
Crucially, these vaccines are manufactured in a water and oil emulsion. And the oil component is just as important as the virus itself.
The oil is that important.
It really is. A classic industry example is the Avipro 105ND vaccine. because the viral antigen inside this vaccine is completely chemically inactivated. It is dead. It functions as an exogenous antigen.
Exogenous, meaning from the outside.
Right. This means the immune system recognizes it as a threat originating from outside the cells. Because it is an extracellular threat, it is ingested by antigen-presenting cells and processed through the MHC2 pathway.
So a different pathway than the vector vaccine.
Completely different. The MHC2 pathway specifically activates the humoral immune system, stimulating B cells.
B cells.
Yes. These B cells act as biological antibody factories, ramping up to produce massive circulating volumes of IgY antibodies.
Wow, and the oil emulsion is really the critical engineering mechanism here, which brings us to our 8th vocabulary word.
A very cool concept, too.
The term is depot effect.
Depot effect.
The depot effect occurs when a vaccine is physically trapped inside a thick, oily bubble under the bird's skin, which forces the medicine to be released very slowly over an extended period of time. To repeat, depot effect.
It changes how the immune system perceives the timeline of the threat.
Yeah, and a practical poultry farming example is the thick oil in the killed vaccine created a strong depot effect, providing the hen with a steady, continuous release of immunity for months on end.
It's the only way to get longevity out of a killed vaccine.
And closely tied to this process is our 9th vocabulary word. The term is seroconversion.
Seroconversion.
Seroconversion is the specific point in time when the bird's immune system has successfully reacted to a vaccine and produced a measurable protective amount of antibodies in the bloodstream. Once more, seroconversion.
It's the proof that the vaccine actually worked.
Exactly. For a practical poultry farming example, we drew blood samples at week 4 to confirm that seroconversion had occurred, ensuring the flock was finally protected.
Right. And to detail exactly how that oil creates the depot effect, we have to look at the injection site.
What happens under the skin?
When a farm worker injects a water and oil emulsion vaccine subcutaneously, the highly viscous oil physically remains at the injection site.
It just sits there.
Yes, and it induces a mild, localized inflammatory response, creating a small granuloma or a tiny lump under the skin.
Okay.
The oil tightly traps the dead viral antigen within this lump. Rather than the immune system clearing the dead virus in a few days, the oil allows only microscopic amounts of the antigen to leak out at a time.
Just a tiny drip.
Exactly. This slow, continuous release drip feeds the immune system over a period of many weeks or even months.
It functions exactly like a slow-release fertilizer pellet you might push into the soil of a potted plant,
That's a perfect visual.
You don't flood the plant with nutrients all at once. The pellet constantly drips a tiny, manageable amount of nutrition into the soil every time you water it.
Right.
The killed oil vaccine does this with immunological training information. It constantly drips a tiny trace of the dead virus to the immune system.
And because the B cells are constantly being stimulated by this slow drip, the immune system never assumes the threat has passed.
It's always on high alert.
Exactly. It never drops its guard, maintaining those high antibody titers for the entire 80-week laying cycle.
That analogy captures the mechanism perfectly. And an important nuance highlighted in the literature is the specific choice of the viral strain used inside these killed vaccines, right?
Yes. Strain selection is vital. High-quality manufacturers deliberately utilize very gentle lentogenic strains, such as the B1 strain.
A gentle strain, but it's dead anyway, right?
It is dead and it cannot replicate, but the physical structure of a gentle strain is vastly preferable.
Why is that?
If you used a killed velogenic strain, the toxic nature of its proteins combined with the heavy oil would cause severe necrotic tissue damage at the injection site.
Oh, so it would physically hurt the bird a lot more?
Yes, it would be a massive, painful lesion. By pairing a gentle B1 strain with a high-quality, slow-release oil, you avoid causing a massive, painful reaction that would stress the bird. Yet you still successfully train the immune system to build that massive, cross-protective wall of antibodies.
That is fascinating. So we have the advanced vector vaccine engineered to build cellular killer T cells, and we have the thick, oil emulsion killed vaccine designed to build the massive wall of circulating blood antibodies.
Two very different, very powerful tools.
Right. And knowing these two powerful tools exist leads us directly to a major controversy in poultry management, the great hatchery debate.
Oh, yes, the hatchery debate.
It brings up the fundamental question of why, in biology, more is not always better.
It really does. The controversy stems directly from fear, to be honest.
Fear of the virus.
Right. Because the velogenic genotype, the 7th virus, is so rapid and so destructive, farm managers operate under intense pressure.
I can't even imagine the stress.
It's high. They naturally want to afford their chicks every possible layer of protection the absolute second they hatch.
Which makes sense on paper.
It does. This fear leads them to over-design their vaccination schedules. They instruct the hatchery to administer both the live recombinant vector vaccine and the thick oil emulsion killed vaccine on day 1.
They do both on the very first day.
Yes. They inject everything into a tiny, day-old chick simultaneously.
However, the academic reports we are reviewing deeply criticize this aggressive day 1 stacking practice.
They tear it apart, honestly.
They note it is particularly disastrous for broiler chickens, which are raised specifically for rapid meat production.
Yes, broilers suffer the most.
Before we examine the biological failures of this strategy, let us introduce our final vocabulary word. The term is vaccine take.
Vaccine take.
Vaccine take refers to the successful attachment and initial multiplication of a live vaccine within the bird's body, serving as clinical proof that the vaccine is actually functioning as intended. To repeat it one last time, vaccine take.
You always want a good take.
Exactly. A practical poultry farming example you would use is, a few days after the spray vaccination, we closely monitored the chicks to ensure a good vaccine take, which confirmed their respiratory immune systems were properly activated.
Without a good take, you have no protection.
Right. So when managers demand both injections on day 1, what goes wrong?
They trigger a cascade of biological failures, and the first of which is severe MDA interference.
Maternal-derived antibodies interference.
Right. We established earlier how maternal-derived antibodies, or MDAs, circulate in the chick's blood. And we know the vector vaccine safely hides inside the cells.
Yeah, it uses the intercellular bridges.
But the killed oil vaccine is entirely extracellular. It flows freely in the tissue space.
Oh, so it's completely exposed.
Totally exposed.
Yeah.
If you inject that killed vaccine on day 1, the highly active maternal antibodies will instantly recognize the exogenous antigen, swarm it, and completely neutralize the dead virus before the chick's own B cells even have a chance to interact with it.
So the mother's antibodies just wipe it out immediately?
Instantly. The vaccine is utterly destroyed by the mother's immunity, making the expensive injection a complete waste of capital. The anticipated seroconversion will simply fail to occur.
So the mother's inherited immune guards essentially cannibalize the killed vaccine.
Cannibalize is a good word for it, yeah.
That is a massive biological waste. But what is the secondary failure cascade you mentioned?
The secondary issue is the immense metabolic cost imposed on the bird.
Metabolic cost? How so?
Well, the oil adjuvant in the killed vaccine is specifically designed to provoke localized inflammation. That is its actual function.
Right, to create the granuloma.
But a day-old chick is incredibly fragile, weighing merely 40 to 50 grams. Inducing severe inflammation triggers a systemic acute phase response.
The chick gets sick from the vaccine.
Exactly. The chick develops a fever and experiences localized swelling and pain. And to fight this intense vaccine-induced inflammation, the chick's metabolism must rapidly reallocate its resources.
It has to shift energy away from growing.
Precisely. It diverts adenosine triphosphate ATP, which is cellular energy, and highly valuable dietary amino acids away from essential developmental pathways.
Like muscle growth.
Right.
Yeah.
Instead of utilizing that energy and protein to rapidly grow skeletal muscle or to lengthen the absorptive villi in its intestinal tract, the chick burns those resources fighting the oil.
Wow.
This severely depresses the bird's feed conversion ratio, or FCR.
FCR is everything in the broiler business.
It is the only thing. For a broiler chicken, where profit margins depend entirely on maximum growth in minimum time, stunting early intestinal development is an absolute economic disaster.
I can clearly see the biological and economic pitfalls, particularly for meat birds. But, I can also understand the operational logic from the farmer's perspective.
Of course. It's about labor.
Right. Catching and handling thousands of birds in a barn weeks later is highly stressful for the flock and incurs massive labor costs.
Huge labor costs.
If you need both shields eventually, it feels intuitively correct to administer them both at the hatchery when you already have the bird in hand. If they are different technologies, why can't the chick's body just process both?
That operational logic ignores the physical limits of the immune system. We call this phenomenon target cell limitation, or take interference.
Take interference. How does that physically happen?
Think about the physical injection. If a hatchery worker injects the cell-associated vector vaccine and the highly viscous oil vaccine into the same general subcutaneous space on the chick's tiny neck, the two vaccines physically and biologically interfere with one another. The thick, sticky oil can physically encase and trap the cells carrying the vector vaccine, preventing them from migrating and spreading the cellular immunity.
It just glues them in place.
Exactly. Furthermore, the intense inflammation caused by the oil attracts aggressive macrophages in a hyperreactive state.
Oh, the Pac-Man cells again.
Right. These localized macrophages are in a frenzy to clean up the oily debris, and in the chaos, they will mistakenly engulf and destroy the host cells carrying the live vector vaccine.
So the cleanup crew accidentally throws away the good vaccine?
Yes. The cellular vaccine take is completely ruined by the local inflammatory response to the oil.
So in your desperate attempt to hold up two different shields at the exact same time, you end up dropping and breaking both of them.
Precisely. The field trial data in these reports is unequivocal on this point. More is definitely not better when administered simultaneously.
Stacking vaccines is just a bad idea.
It risks severe physical and immunological interference. Now, if a veterinary protocol absolutely mandates that both must be given at the hatchery, if there is no other way, they must be spatially separated.
You mean injected in different spots?
Yes. They must be injected in two completely distinct locations, such as the extreme opposite sides of the chick's neck, to prevent local physical interference.
But even then,
With spatial separation, the heavy metabolic cost of the oil inflammation on the day-old chick remains a severe negative factor.
if cramming everything into day one is biologically flawed, what is the optimal sequence?
The optimal sequence is a phased approach.
How do we build these three critical layers of protection, mortality, respiratory, and reproductive without destroying the bird's early growth curve? This brings us to the final synthesis of our sources, the gold standard program, the ultimate architectural blueprint.
Yes. Synthesizing all the virology and immunology we've discussed, the academic sources detail a highly orchestrated, multi-platform blueprint designed specifically to protect long-living layer or breeder flocks against velogenic genotype 7 strains.
Okay, so this is for the layers and breeders.
Right. It is a carefully phased, four-step process that respects the bird's developing biology.
Let us walk through the timeline. Step 1 occurs on day 1 at the hatchery. What is the precise protocol?
On day 1, the focus is strictly on establishing the foundational base. The chicks are injected with the RHVT-ND vector vaccine.
Okay, the vector vaccine goes in.
Yes. This immediately begins the slow process of building the lifelong cell-mediated immunity, deploying the CD8 plus killer T cells to patrol the tissues.
And what about the oil?
Crucially, they completely avoid the heavy oil injection. Instead, they administer a live lentogenic vaccine via a coarse spray.
A spray.
Yes, the chicks literally inhale this gentle live virus. Because it is inhaled, it bypasses the circulating maternal antibodies in the blood and immediately begins colonizing the respiratory tract.
So the mother's antibodies can't touch it because it's in the lungs, not the blood.
Exactly. This stimulates rapid local mucosal immunity without inducing a systemic fever or stunting the chick's metabolic growth.
That is brilliant. Okay, moving to step 2, which is the rearing phase. The birds are growing over the next couple of months. What is the management plan during this developmental window?
During the rearing phase, the focus is on maintaining and expanding that mucosal shield. Farm managers will administer mild live vaccines via aerosol spray or through the flock's drinking water lines every few weeks.
So they just keep hitting them with mild exposures.
It is a critical maintenance step. Mucosal immunity is highly effective, but it is short-lived. By exposing the respiratory and intestinal tracts to the live virus repeatedly, they're constantly topping up the local defenses.
Keeping the guards awake.
Exactly. Ensuring a thick, robust shield of secretory IgA antibodies remains active in the throat, lungs, and gut.
Then we reach step 3, the prelay phase, when the pullets are roughly 14 to 16 weeks old. They are fully grown and are just weeks away from initiating egg production.
This step is the absolute masterstroke of the entire immunological program. This is the exact moment they introduce the inactivated oil emulsion vaccine via intramuscular injection.
Week 15, why is the timing so perfect?
The timing is biologically perfect because by week 15, the maternal antibodies inherited from the mother are completely exhausted and gone.
Ah, so there's no MDA interference.
Exactly. Absolute zero MDA interference to destroy the killed vaccine. Furthermore, because the bird has been repeatedly exposed to the live spray vaccines during the rearing phase, its B cells are already highly primed and sensitized to the Newcastle antigen.
They've been practicing for 15 weeks.
Right. When you inject the concentrated, slow-release oil vaccine into this fully mature, heavily primed immune system, it triggers a hyperreactive biological explosion of antibodies.
An explosion.
The circulating blood volume becomes completely supersaturated with IgY. The viremia barrier is locked down tight, providing overwhelming numerical superiority against any genotype of the 7 virus attempting to enter the bloodstream.
It is genuinely fascinating how perfectly timed that is. It's like choreographing a biological dance.
Huh.
And finally, we have step 4, the production phase. The hens are actively in the barns, laying eggs every day for the next year or more.
During the production phase, the manager must never forget the anatomical vulnerability we discussed earlier, the lung oviduct
The
access.
secret tunnel.
Exactly. The blood is saturated with antibodies, thanks to the oil vaccine, but as we noted, localized mucosal immunity degrades quickly over time.
So what did they do?
To ensure that secret air sac tunnel remains permanently sealed off, the manager must top up the flock with a live mucosal spray vaccine every 6 to 8 weeks throughout the entire laying cycle.
Every 6 to 8 weeks, just constantly renewing it.
This ensures the physical barrier protecting the reproductive organs remains completely impenetrable, preventing the virus from bypassing the blood guards.
I am truly struck by the profound elegance of this 80-week timeline. I mean, it replaces the panic-driven instinct to just inject every available medicine on day 1 with a highly choreographed, deliberate plan.
It's science applied perfectly.
It respects the decay of maternal antibodies, utilizes the vector technology to hide from interference, primes the B cells with mucosal sprays, and triggers the massive oil-adjuvanted explosion exactly when the reproductive organs need the highest level of protection. Every single intervention has a precise, scientifically validated reason.
It really is the pinnacle of applied veterinary immunology. And if we look at the broadest implications of this strategy, there is one final compounding benefit, particularly for primary breeder flocks.
Breeder flocks. These are the highly valuable birds producing the fertilized eggs that will become the next generation of chicks, right?
Exactly. Because this gold standard program pushes the hen's circulating blood antibodies to such extraordinary heights, a biological transfer occurs.
What kind of transfer?
The mother hen actively packages those massive volumes of IgY antibodies and secretes them directly into the developing yolk of her eggs.
Oh, wow. So she passes the wall on to her babies.
By achieving hyperimmunization in the mother, you guarantee that her offspring hatch with the highest possible concentration of maternal antibodies, giving the next generation an incredibly robust shield as the entire cycle of protection begins anew.
That is an incredibly satisfying biological loop. Well, we have covered a massive amount of dense scientific ground today.
We really have.
We detailed the precise cellular pathology shutting down the reproductive organs, exposed the anatomical secret tunnels the virus exploits, and constructed the ultimate multi-platform vaccine blueprint to defend the flock.
It's a lot to take in.
It is. So, before we close the deep dive, for our listeners meticulously practicing their Ingalls shadowing, I am going to do a rapid, clearly pronounced recap of our 10 critical vocabulary words. Listen closely and repeat one final time. Genotype, viremia, apoptosis, mucosal immunity, shedding, maternal antibodies, vector vaccine, depot effect, seroconversion, vaccine take.
As we conclude this extensive review, I want to leave you with a final, somewhat provocative thought that extends naturally from the pathology we explored today.
I love a provocative thought. What is it?
Well, we have detailed how highly evolved viruses like Newcastle disease have developed mechanisms to actively exploit the specific anatomical quirks of their hosts, specifically utilizing the direct physical tissue contact between the avian air sacs and the oviduct to create a secret tunnel that bypasses systemic immunity entirely.
Right, the lung oviduct axis.
Exactly. If pathogens are exploiting physical anatomy to this degree, it forces us to ask a larger structural question about the next frontier of veterinary science.
Which is?
Will the future of vaccine technology need to move beyond simply saturating the blood plasma and coating the mucosal surfaces? Will bioengineers eventually need to figure out how to specifically program and anchor immune cells to physically guard the exact anatomical bridges that connect vulnerable organ systems?
That is a staggering concept. Moving beyond circulating guards to engineering stationary garrisons permanently stationed at the biological secret tunnels themselves, it shows just how much further immunology might evolve. Thank you so much for joining us on this incredibly detailed deep dive today. We hope the deliberate pace aided your shadowing practice and that you walk away with a much deeper understanding of both the vocabulary and the profound biology of poultry management. Until next time, keep questioning the science and keep learning.